In this video, we will discuss about the importance of N-glycosylation of TGF-β receptor 2, which is usually expressed on the cell surface, and how if this N-glycosylation of TGF-β receptor 2 is affected, then it will also limits the TGF-β1 effect. So basically, TGF-β1 helps in the EMT, that is epithelial-to-mesenchymal transition, and in that case, the smad pathway plays an important role. And also this TGF-β receptor 2 plays an important role, because from that it will trigger the smad pathway. So the hypothesis that paper tested is this N-glycosylation of TGF-β receptor limits the TGF-β1 effect. So first they took some cell lines and checked whether those cell lines are responsive to the TGF-β1 or not. For that they used this CAGA luciferase receptor. So they first transiently transfected this CAGA luciferase receptors and then treat them with this TGF-β to see whether TGF-β can enhance this luciferase signals or not. And out of the many cell lines, 2 cell lines does not give any response, that is MKN45 and MKN28 does not give any response. However, other cell lines, that is lung cancer, the other cell system, that is HeLa cells, shows a significant upregulation of the CAGA luciferase in the presence of TGF-β, indicating those are TGF-β responsive cell lines. Thereafter, they checked them using the TGF-β. They checked them using the TGF-β receptor 2 conjugated with the flag tag antibody. So what it did is they knock in the flag tag at the C-terminus of Tβ receptor 2 and then check the flag tag that would give them indication about the Tβ receptor 2, because when they did their experiment they did not find a good antibodies against this Tβ receptor 2. So they used the flag tag to find out the amount of these Tβ receptors, and they are able to see different molecular weights indicating that Tβ receptors could have the different N-glycosylations or other glycosylations. So far they just confirmed that the cell lines that they are using are TGF-β responsive, and they will also enhance the Tβ receptor 2 expressions. Now they wanted to check the impact of N-glycosylation. So they used 2 different inhibitors that works in 2 different ways. First one, that is KIF, helps this N-glycosylation precursor to attach to the protein backbone or peptide backbone, we can say that. And in the case of TUN inhibitor, it does not allow the N-glycosylation precursor to attach to the peptide backbone. Precursor is mannose 9 glucose 3 and N-acetyl glucosamine 1. So this is the N-glycosylation precursor, which are usually found in the endoplasmic reticulum, where it attached to the peptide backbone. So those things, in the case of KIF, it is attached, but the further N-glycosylation gets inhibited, and in the case of TUN inhibitors, this N-glycosylation precursor does not attach to the peptide backbone. So what they are able to see that they check them using the CAGA luciferase signals in the panel C, and they are able to see that in the KIF inhibitors there is not significant decrease in the CAGA luciferase. However, when they use the TUN inhibitors, they are able to see a significant decrease in the CAGA luciferase, indicating that TUN inhibitors affect the N-glycosylation more, and that will cause the that will affect the Tβ receptors to interact with this TGF-β. And later on they also check them using the endogenous Tβ receptors antibodies, and here also they see the similar results, that is smad signaling pathways goes down when they use the TUN inhibitors, and when they check it with the flag antibodies, here also they see the similar results. And they also see that the size of the flag tag or Tβ receptor 2 is slightly lower than they used it for when they used for KIF, because in the KIF the precursor N-glycan precursor attached to the protein backbone, but in the case of the TUN inhibitors, the N-glycan precursor does not attach to the protein backbone. So the expected weight that they have mentioned in the paper is that when this TUN inhibitors is used, they are predominantly able to see more of 64 kilodalton size Tβ receptors too, and when they used these KIF inhibitors, they are able to see 68 kilodalton TGF-β receptor 2 size. So, and these things are quantified on the bottom side of this panel D and E. Thereafter, the hypothesis that comes into a mind that when the N-glycosylation gets affected, then this Tβ receptors 2, whose usual location is on the cell surface, does not able to express on the cell surface. So they might be perinuclearized, they might be in the perinuclear region. So in order to check that, they use the confocal microscopy, and there in the case of control, they are able to see that most of this flag tag, they checked it using the flag tag, are expressed on the cell surface. And for the case of KIF, some of them are internalized, but still most of them are on the cell surface. But when they check it with the TUN inhibitors, they are able to see that there is a huge concentration of this flag tag or this Tβ receptors 2 in the perinuclear region. So this in all together indicate that these N-glycosylations are important for this TGF-β related effect, trigger SMAD pathways, and also when the N-glycosylations get inhibited or disturbed, and there is a this cell surface expression of this Tβ receptor 2 gets affected, and they might get stuck in the perinuclear region, and because of that, the downstream signaling pathways, that is SMAD pathways, will not be triggered as efficiently as in the case where Tβ receptors 2 are normally glycosylated. This is what they finally come to the end from the results that are presented in this figure. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.